Next up is Victor Ruiz. Victor is a biology major at UC Berkeley and is also expecting to graduate in May 2020.
Today I'll be presenting my research on TP53 mutations in the development of high-grade serous ovarian cancer, serous being the general type of epithelial cell this cancer is derived from. I decided to conduct research on this topic because not only is cancer the second leading cause of death worldwide, but as a research scientist, I want to be able to eventually help cure disease such as this one and better understand the molecular processes. The topics I'll be covering include normal functioning P53, also known as WTP53 or wild type P53, and how it assists in tumor suppression. Next, I will be describing the development of mutant P53 and how this can lead to cancer. After we understand how TP53 can lead to cancer, I will then discuss the classification and origins of high-grade serous ovarian cancer. I will then cover the methods of treatment and detection for high-grade serous ovarian cancer and the development of cisplatin resistance from these treatments, along with future possibilities that researchers can look at in order to combat this development. So, wild-type P53 is the name of the gene that encodes the protein P53. This protein is commonly referred to as a guardian of the genome due to its ability to maintain genomic stability. The encoded protein is composed of 393 amino acids that can make up 4 general domains. These 4 domains are the transactivation domain, proline-rich domain, DNA binding domain, and the tetramerization domain. Each of these domains are composed of a specific structure that gives them their function. Now, the utilization of these 4 domains provides P53 with tumor suppression capabilities via several complex processes that result in the cell cycle arrest, autophagy, apoptosis, and regulation of P53. So, how common are these mutations of TP53? Well, in the image on the shown, several tumor sites were tested for the presence of TP53 mutations. And as we can see, mutations were found in nearly every cancer from rates of 5 to 50%. And they can become even more frequent as cancer advances. Therefore, TP53 mutations are quite common in cancer. Furthermore, these mutations are most common in ovarian cancer, which is why I focused on this topic. So what causes these mutations? Well, like any mutation, TP53 mutations can be induced by spontaneous mutations, DNA repair errors, and induced mutations. However, if there is a mutation of TP53, then the chances of cancer can now be more than 90%. In the image, the TP53 gene is shown. And along this gene, there are several mutation codes extending vertically. The exons 5 through 8 in red represent the DNA binding domain, which is the most commonly mutated part of the gene. Now let's talk about how these mutations can lead to cancer. Mutant P53 can display several detrimental effects, such as the loss of function, dominant negative effect, and gain of function. Loss of function mutations can destroy or create irregular structure for the protein and leads to its inability to interact with DNA. While dominant negative effect has a sort of zombie effect that results in the mutated P53 stopping the wild type P53 from properly functioning or forming at all. These 2 effects can lead to the loss of tumor suppressing abilities by TP53, whereas a gain of function can be either the suppression or exaggeration of wild type P53 functions, and in both cases, this initiates the formation of tumors. Now, nearly all cancers have shown TP53 mutations, as previously stated, and the most common is ovarian cancer, where TP53 mutations are found in 95% of all ovarian tumors. Epithelial ovarian cancer accounts for 90% of these cases. Epithelial ovarian cancer has been further divided into type 1 and type 2 tumors via the dualistic model. Since type 2 tumors make up 90% of ovarian cancer deaths and are even characterized by their presence of TP53 mutations, I decided to focus on this type. Okay, so in order to understand how we can tackle this form of cancer, we must first understand where the cancer forms and why it forms there. The origins for the initiation of high-grade serous ovarian cancer include the ovarian surface epithelium, the fallopian tube epithelium, and both of these describe the uptake of one or more of these stressed epithelial cells provides a environment that helps initiate the growth of a tumor. So now that we understand the initiation and origins of high-grade serous ovarian cancer, let's talk about some of the treatment methods. First off, early detection of high-grade serous ovarian cancer is at a huge disadvantage. This is due to its lack of screening strategies and lack of absence of early symptoms. Only about 13% of high-grade serous ovarian cancer patients are diagnosed in stages 1 or 2. The 10-year survival of patients diagnosed in an advanced stage is 15%. And when symptoms arise, the disease is found to be in an advanced stage in about 75 to 80% of patients. Some of these symptoms include abdominal pain, bloating, nausea, constipation, fatigue, back pain, and elevated urinary frequency. After cancer has been detected, the first step in treatment is surgical excision. For stage 3, this involves a spread of cancer to organs and tissues within the peritoneal cavity and retroperitoneal lymph nodes, while stage 4 includes a spread to areas outside of the peritoneal cavity, such as the lungs. Surgical excision is the first step of treatment. However, it is not the last. The next step is platinum taxane combination chemotherapy. Cisplatin is the platinum compound and paclitaxel is the taxane compound. Both of these are used to slow neoplastic proliferation. This method is effective as a first-line treatment. However, 75% of high-grade serous ovarian cancer patients experience a relapse. Recurrent high-grade serous ovarian cancer patients typically develop resistance to chemotherapy and unfortunately succumb to the disease. Within the image, the interaction between cisplatin and DNA is shown. This interaction is very important because it allows for the inhibition of cell division and mitochondrial respiration, as well as the induction of apoptosis. In other words, this helps decrease the growth of tumors. So what's wrong with it, right? Well, cisplatin can become resistant within the cell. In order for the cisplatin to do its job, it must enter the cells. However, the degradation of copper transmembrane proteins, CTR1 and CTR2, leads to the development of cisplatin resistance by decreasing the cell's uptake of cisplatin. However, these are not the only contributions to cisplatin resistance. P53 has a large role in the determination of cisplatin resistance. Both wild-type P53 and mutant P53 can regulate cisplatin-induced cell death. It has been associated as a determinant in the chemosensitivity of high-grade serous ovarian cancer. This suggests that the mutant P53 may induce tumorigenesis while simultaneously increasing the resistance to the chemotherapy drug. It can do this by losing its ability to transactivate Bax gene, hence increasing the proliferation of high-grade serous ovarian cancer and its resistance to chemotherapy. Therefore, cisplatin-resistant cells not only lead to higher levels of mutant P53, but also increase in oncogenic activity and have a higher risk of cancer relapse and death. One specific mutation that leads to the resistance of cisplatin is P.R175H. As previously mentioned, mutations can cause loss of tumor suppression function as well as gaining oncogenic abilities. This protein formed from the mutation, which seemingly gains a function in promoting cell survival and protecting cells from damage induced in chemotherapeutic cell death. As shown in the picture, caspase-3 is a key component in the normal induction of apoptosis. However, this mutation blocks caspase-3 expression, thus promoting tumor growth. The mechanism by which this happens is shown in the image. All right, so what can we do about this, right? Possible future research should be done on the inhibition of glutamine metabolism. Glutamine metabolism was found to be substantially higher in cisplatin-resistant cells when compared to cisplatin-sensitive cells. As the glutamine concentration remained constant, fewer cisplatin-resistant cells could be generated than cisplatin-sensitive cells. Rapid glutamine catabolism hindered the growth of cisplatin-resistant cells due to excessive glutamine consumption and providing insight into a future chemotherapy research. However, the inhibition of glutamine metabolism does not come without its consequences. Glutamine is a preferred source of energy for intestinal mucosa, and the high levels of glutamine are required for proper immune system activation. Kidney cells utilize glutamine for acid-base balance, as well as production of ATP, the energy for the cell, and via the tricarboxylic acid cycle. Let's talk about the mechanism of inhibition of glutamine metabolism. Cancer cells utilize glutamine for energy production in order to satiate quick cancer cell division and expansion. When glutamine availability was decreased in cisplatin resistant cells, they displayed a 40% decrease in cell viability, whereas sensitive cells displayed only 11% decrease. Therefore, resistant cells are much more glutamine dependent than non-resistant cells. Okay, so how can we go about decreasing the glutamine metabolism of a cell? Well, glutaminase and glutaminase 1 are isoenzymes that provide glutamine to the cell. Two possible compounds that can inhibit these enzymes from producing glutamine are 968 and BPTES. 968 can prevent an inactive glutaminase from undergoing a conformational change. This inhibits its ability to produce glutamine. While BPTES, on the other hand, can inhibit an active glutaminase 1 from producing glutamine, as well as resensitizing cisplatin resistant cells to the cytotoxicity of the platinum compound. While both of these have many great benefits, they do not come without their shortcoming. 968 is unable to inhibit the activity of an activated enzyme tetramer, and BPTES is highly hydrophobic, thus hindering its utility as a pharmacological probe. For future research to be done, we need to first find a better inhibitor of the GLS and GAC enzymes. And that concludes my presentation.